How do new genes evolve? As we've learned in previous animations, a gene is a long stretch of DNA containing information that codes for something, usually a protein or a group of proteins. Point mutations can edit small bits of information within a gene, modifying the type of protein that it builds. These small edits are extremely important for evolution, but if we compare the genes of, say, a flower to those of a dolphin, we see that even though they do have many genes in common, dolphins also have entirely new genes that flowers do not have. Flowers also have genes that can't be found in dolphins. This observation forces us to ask the question, how do entirely new genes evolve? Point mutations are clearly not enough. Well, it turns out that over the years, scientists have discovered many natural mechanisms for the evolution of new genes. One of the most common and well-understood pathways is a duplication event followed by further mutations. A duplication event is a special type of mutation where, as you may have guessed, a stretch of genetic code is duplicated and reinserted into a creature's DNA. Duplications happen naturally all the time. They can be small, a few letters or nucleotides long. Other times, entire genes can be duplicated, dramatically increasing the length of a creature's genetic code. During duplications, and in the generations that follow, further mutations can occur in the new gene, giving rise to entirely new genetic information that codes for new proteins with new functions. Scientists have directly observed duplication events in the lab many times over. Because of this, they can now examine the genetic code of any living creature, look for known signatures of past duplication events, and begin to piece together an understanding of how specific genes likely evolved. Here we'll look at 3 traits which arose from gene duplications: the dachshund's odd-shaped yet powerful legs, the unique digestive enzymes of leaf-eating monkeys, and finally, the evolution of snake venom. At first glance, you might be tempted to think that the dachshund's short legs are simply a disability. To the contrary, the unique shape of this dog's legs make it a surprisingly powerful digger, and most importantly, allow it to enter small burrows to coax out rabbits, groundhogs, and even badgers from their dens. Other dogs can only dream of such adventures. By looking at the dachshund's DNA, researchers have found that their unique legs are the result of a duplication event. A gene called FGF4 was copied and inserted elsewhere in their DNA. The new gene happens to produce protein in a way that interacts with their growing bones, reshaping the dog's legs and opening up an entirely new hunting niche for the animal. Humans who liked the trait bred the original dog with many others, eventually giving rise to several new dog breeds and proving that sometimes, even the strangest of mutations within the right environment can turn out to be extremely beneficial. Now let's look at the fascinating case of a leaf-eating monkey from Vietnam, the Duc Longour. Several species of Asian monkeys eat almost nothing but leaves, a diet that would cause humans and most other primates major stomach problems. The monkeys achieved this amazing feat of digestion with the help of several adaptations, one of which was first made possible by a gene duplication. RNase-1 is a protein found all throughout your cells and blood. Experiments have shown that this protein helps our cells fight against viruses by attaching to and breaking down virus genes. In your intestines, RNase-1 does a similar task but for a very different purpose. There, it breaks down genes from the cells of the food you've been eating, converting those genes into nutrients that your body can absorb. In humans, the pH or acid levels of your intestines are pretty much the same as the pH levels of your cells. This allows a single version of RNase-1 to work great fighting viruses in your body and digesting food inside your intestines. In leaf-eating monkeys, however, the intestines are more acidic. This acid appears to be helpful for breaking down the tough cell walls of raw leaves, but unfortunately, the extra acid also slows down RNase-1 proteins, which are extremely sensitive to acid. Scientists have found that a relatively recent duplication of the monkey's RNase-1 gene has fixed this problem. The original gene still makes normal protein to help fight against infections, but the new gene, after being duplicated, began accumulating mutations that slowly made it better and better at functioning in acid. Here we have a clear case of a single gene that was once okay at 2 separate jobs, was then duplicated, and the 2 genes have since specialized to produce proteins for different tasks. Now for an exciting yet slightly disturbing example, the evolution of snake venom. Genes inside the saliva glands of most creatures, humans included, produce special proteins that are able to start breaking down food on a chemical level even before it gets to the stomach. Venomous snakes, however, have taken this a step further. Their saliva glands produce venom, a cocktail of proteins and other molecules that kill their prey when injected. Let's see how one of these deadly proteins evolved. Many people assume that blood clots form when cuts are allowed to dry in open air. Amazingly, clots actually form through a series of chemical reactions that can quickly seal a wound, even underwater. This ability is possible in part because of a protein called factor X. It's found in the blood of many animals including fish, frogs, snakes, birds, and people. Factor X normally exists in a dormant or sleeping state, drifting about the bloodstream with no effect. If a blood vessel is cut, however, chemicals of the damaged tissue activate factor X at the scene of the injury. Factor X then initiates a series of chemical reactions, causing a clot to form and seal the wound. The saliva of the Australian rough-scaled snake is loaded with pre-activated factor X proteins. If the snake bites an animal, injecting its saliva directly into the wound, rapid clotting occurs throughout the victim's body. The result is often death. When scientists look at the genes that code for this venom, they see clear signs that the snake's very own factor X gene, the one that it uses in its own blood, was copied through a duplication event. After, or possibly during duplication, mutations in and near the new gene caused it to produce factor X protein in the venom glands instead of the blood. As time went on, small mutations built up in the duplicated gene, making it more and more efficient at its deadly new task. Here we see that a gene once used for healing has now evolved to kill. So to sum things up, how is it that new genes evolve? One of the most important and well-understood pathways is for genes to duplicate and then accumulate new mutations. Gene duplications, in combination with simpler mutations like insertions, deletions, and point mutations, are happening naturally, right now, all throughout the living world. With these mutations constantly occurring and constantly filtered through natural selection, there are no limits to the variety of new genetic information, new traits, and new species that evolution can produce.